start this session. So welcome everyone. My name is Barbara Wong, a PhD student at Université Laval, and I'll be your moderator for session 10, our last Q&A session for Forest Health. Our first question is great talk. So for everyone that's joining in, please feel free to submit a question in the Q&A box and I'll distribute them accordingly. So our first question is for Barley. What will it mean if you do not find any resistance in UK Sitka spruce?
So thank you for your question, Hayley. I think that's probably going to be the likely scenario. And that doesn't mean necessarily that there isn't any resistance anywhere. All of the UK Sitka spruce came from Haida Gwaii. And for the white pine weevil, there's no resistance there. So even though obviously they're not the same insect, I'd expect resistance to one to potentially confer with resistance to another. So if there's no resistance in the UK population, that's okay. I'm still hoping to do an RNA-seq study where I look at differences in response to the insect within the UK population, and then I'll do a similar damage response study, but using Canadian Sitka. I hope that answers your question.
And our next question is for Heather. Did you see any difference between the two large species in the field? What does this mean for management?
So although we do have two large species, the majority of it is Japanese larch. Unfortunately, the foresters who do most of the recording don't actually distinguish between which species when they record how badly they're affected. So of the 1,200 reports we have of remorum on larch, only about 100 of them are actually distinguished by species. Of those 100, only 6 are on European larch. So the others are all on hybrid or Japanese larch. So it does seem as if there is some sort of lesser apparency of remorum on European larch, but again, that could just be because we have a lower level of European larch planted, so it's harder to spread between the stands because they're more spread out. In theory, that could lead to a higher level of planting of European larch, but the reason that originally we've got so much Japanese larch is that European larch gets larch canker quite badly, which causes cankers on the stems and makes it pretty much unviable for commercial forestry, which is why everyone's planting Japanese larch instead. So if you switch back to planting European larch in order to get an avoidance of remorum infection, you might end up with canker instead. There was some talk about using other larch species, so western larch or oriental larch, but those seem to also be affected by remorum. So it's an area of study that I'm not doing, but hopefully someone else will carry on and take further.
And the next question is for Mengmeng. Were there any differences between the lodgepole pine, jack pine, and their hybrids in response to Dactystroma infection?
Yes. This experiment was performed by Dr. Nicolas Faux and Dr. Richard Hamlin's group, and they have already published the results earlier this year. And I remember they found hybrid has immediate response between jack pine and the lodgepole pine, and jack pine showed higher resistance. Or sorry, I didn't remember this result clearly, but I can confirm, yes, they have already published this result.
I can speak on this because I helped with the work. We found that jack pine had a higher level of susceptibility to Dactystroma compared to lodgepole pine. But yeah, the next question is for Barley. How did you analyze your cellophane images? It seems ideal for machine learning.
I used a pretty simple piece of software. I used ImageJ, and you can just create.
I'm sorry, can you repeat that? You kind of cut out.
I used ImageJ, so the cellophane images were just red or white, and so it was just with a very simple cutoff for what the pixels were, and it took the image, drew circles around all of the red patches for me, worked out the average of those, which circle was the biggest, and the total area for each image. So yeah.
And then the next question is from Mengmeng. What do you think this means about the importance of protein coding versus gene expression variation and resistance? This is in response to your results of DN over DNS and expressions.
Your results are really interesting. What do you think this means about importance? Okay. I think this means we found some positive selection markers in this resistance mechanism. That means this resistance can be heritable and maybe durable because it was accumulated a long time in the past. So this is a good sign if we want to use this mechanism for future species improvement.
Awesome. And then our next question is for Eddie. How many other genotypes like 4 and 9 are in the NCSU Rust improvement population?
That's a good question. It's actually very rare to find parents with this sort of multi-environmental resistance. Most parents show a lot of rank changes. If you grow their progeny across 10 environments, you'll see all kinds of changing of ranks. But only a very small number, maybe less than 10, show this very high degree of rank stability across environments, no matter where they're planted. So that, yeah, I think these sorts of general resistance genes are rare in the population.
And then the next question, I think this is for Heather. In larch, have there been any resistance trees confirmed of either larch?
Well, that's actually chapter 3 of my thesis. In the UK, we currently have a felling policy. So anything that's infected gets felled, and all the larch within 250 meters around that also gets felled. So it's not very good for finding survivors or resistant trees. However, there are 2 management zones where felling isn't immediately ordered. And within those areas, we've managed to identify some survivor trees, which are ones that survived in stands of pretty much 99.9% mortality. There'll be one tree on a hillside that survived. So we've taken scion material from that, we've grown it up as grafted trees, and we have carried out tests by inoculating excised material. So although all of our trees did form lesions in response to infection, there was a significant difference between the most susceptible and the most resistant or tolerant individuals. So in theory, there is some difference within the population. And if you were to carry this on further, then we can do an ad project and look at the ones with the smallest lesions. You could then use that as a resource for looking at further resistance within the larch. So there is some variation in the population, and there may be resistance or tolerance, but we're still working on that at the moment.
And the next question is from Mengmeng. Your correlation was moderate between expression levels and DNDS in DGs. Does this indicate that much more indirect trans effects would exist than direct cis effects?
It's possible, but I can't confirm it right now because I think it needs more experiment to confirm trans and cis effects for my current transcripts. With these RNA-seq results, I'm reluctant to say these are trans, these are cis, but I would like to test these hypotheses sometime. And I think it's also very good for jivas if we have these trans and cis effects. Yes, thank you.
The next question is for Eddie. In your inoculum, have you included many strains? If yes, how many and from which regions?
Yes, the inoculum was composed of 150 different galls, different gall isolates, and they were taken from the highest rust incidence areas in the southeast United States. So that included South Carolina, so southern South Carolina, northern and central Georgia, and part of Alabama, I believe. So they were very wide. It was a very wide-ranging and diverse set of isolates that were combined.
The next question is for Barley. Is there any momentum to incorporate more Sika germplasm into reforestation? It seems like there are many other superior provenances that perform at least as well as the Haida Gwaii material across a larger area.
So most of the seed that's grown in the UK currently is improved Sitka. The Forestry Research, which is the government-run forest research centre in the UK, had a breeding programme for Sitka for a number of years for wood properties. Subsequently is improved seed from that study, but all of the trees that initially went into that study were the trees from Haida Gwaii. So from a commercial perspective, the growers want to grow what they know works in their location. And I think that's why Haida Gwaii was chosen initially when the seed were brought over, due to similar climates, etc. So from what I've seen, no, not really. But I don't think that means it wouldn't happen ever. It's just that the current breeding program is based on old seed, basically.
And then our next question is for Heather. Are you aware of the response of interspecific hybrids between the Japanese and the European larch to the disease since they are often planted?
So although hybrid larch is more extensively planted than European larch, a lot of the seed comes from seed orchards where they just alternate rows of European and Japanese larch and then call that hybrid larch and just presume that the pollen and seed has mixed in the right proportions. So it's quite a variable seed lot and it's not always actually hybrid larch, it can be a mixture of them. Within our research area in the Galloway Forest, there wasn't much hybrid larch available for us to work on, so we didn't include it in any of our research. However, from the SPHNs, so the Statutory Plant Health Notices, which are the records of Phytophthora morum in England, out of the 112 that we have that we know by species, only 17 of them are on hybrid larch. So again, a lesser extent Japanese larch, but it's also planted to a lesser extent. So we don't know enough to be able to answer that question properly, but I'm sure if you actually did some work, you would find out that there might be a difference. But considering there doesn't seem to be much difference between the two parental species, I would suggest it's unlikely that the offspring of the two parents would be that different.
Excellent. The next question is for Eddie. It's from Richard, and he's asking, are the results written up yet? I would be interested in seeing more.
Thanks, Richard. Yeah, that's a good question. And yes, they have been written up in the final rounds of revision now with the journal Heredity. So they should be out, I'm expecting pretty shortly. I think the results are interesting because it shows that these QTL are additive on the liability scale. So, you know, individuals with both resistance alleles were almost immune to the disease. And so it's showing that they have some sort of additive component with one another on the disease liability scale. So it was immunity. I mean, there was one individual that had both resistance alleles that did get disease, but Fikret and I are still wondering if that might have been some sort of an error. So it's harder, you know, we have numbers that small, it's kind of hard to know if they're real or not. But yeah, it does seem like these QTL vary with respect to their impact on liability, and they are additive with one another. So those are pretty important findings from the mixed inoculation experiment, I think.
Does anyone have any more questions? Please feel free to submit them to the Q&A. Well, I have a quick question for Barley. So I know that you measured your area consumed by the gram of the weevils. And I was wondering, is there like a big difference between the size and the age of the weevils that you decided to do that?
So I measured the weight of each weevil that I put in the enclosure, and I put 2 weevils in each enclosure, 1 male, 1 female. Their ages we don't know, so they're collected live off billets rather than kept alive in culture. So I've got no idea the range of their ages. They can live for up to 3 years. They were collected in August, so you'd expect the new insects to be emerging around August. So we were kind of hoping to get younger insects just so we could then keep them alive in the lab for longer subsequently. But no idea how old they were. And with regards to weight, we recorded that and I took that into account, and it's been normalized against weight. So.
The next question is for Eddie. Eddie, are there markers for major fusiform rust-resistant genes like the one found in 10.5?
So the one found in 10.5 was mapped back in 1996, and back then they just had rapid markers, and so those are sort of distantly linked to the gene. So those would be of sort of limited utility now because they're really hard to run, very expensive. You need gels and expensive lab setup. Part of the experiment that I presented in this presentation, I did actually develop some AgriSeq markers. Those are amplicon sequencing markers. It's based on Oxford Nanopore technology. And yeah, we have developed markers that are very tightly linked to the genes. I've also tried to look into the genome at those RNA-seq hits that we got, but these NLR genes are part of highly duplicated gene families, and they're notoriously difficult to work with. So it's going to take more time to develop markers within those candidate genes, but we have markers that are very, very tightly linked, for which there was actually no recombination apparent between the marker and the gene. So we can use those for marker-assisted selection.
And then this is another question for you, Eddie. For the one individual, did you collect spores on the odd chance that it's a virulent or aggressive strain?
No, that would have been interesting. The way I'm kind of thinking about this is maybe the probability of infection is some sort of a multiplicative function of the virulence allele frequencies in the pathogen population. So the fact that an individual with 2 resistance genes is more resistant than an individual with one resistance gene, that kind of suggests to me, well, okay, there's virulence alleles, and some proportion of the population is virulent to A, and some proportion of the pathogen population is virulent to B, and a very, very much smaller proportion of the pathogen population is virulent against A and B. So what it looked like to me is that maybe the probability of infection is multiplicative, and it's a function of the virulence allele frequencies in the population. But until we actually start to sequence the rust isolates and actually isolate spores like Richard suggested, we won't really know.
And then the next question is for Bailey. It's from Jean, and he said, you have been testing and found gall-free Sitka spruce. Do you have information about the geographical origin of these trees? And are you planning to check the level of introgression of these trees with white spruce genes that may occur at the eastern margin of Sitka spruce natural range?
So we've not been looking at galls at all. So the way that the large pine weevil attacks the Sitka is just by eating the bark. And so you see the area of bark that's been damaged. But when I'm bringing seed over from Canada, I'm planting it in the UK, which is what I was talking about with the provenance trial, which I maybe mentioned right at the end of the talk to say is something I'm going to be looking at later. I do know, yeah, where those seeds came from throughout Canada, and I'm going to be looking at that as one of the factors, both longitude, latitude, and elevation. I wasn't going to look at introgression, but it's a good point, and it's definitely something I should also consider, because I do know that that is a thing that happens, and I also know that it affected the white pine weevil studies, so it would make sense that I also looked at that. Thank you, and I'll say hi to John.
And then the next question is for Samson. What implications does your work have for the resistance breeding?
So our results generally would, help us to understand the mechanism for resistance. And so with our results, we can, drill down to a more refined phenotypes that can also enhance, let's say, genomic selection. And for instance, with, the gene expression profile as it differs between lodgepole and jack pine, we can look into, the biology or the function of these genes and, what it tells us with regards to, like, for instance, chemical defenses or protein defenses or, or let's say cellular structure. And so this would also lead us to like more physiological or biochemical study. And so if we understand the general mechanism, it's basically, it helps us to like, identify a more like a better phenotype, that can enhance our genomic selection.
Are there any more questions from the participants? If there's any more last minute questions, please submit them to the Q&A. Otherwise, we could end like 3 minutes early so everyone can catch a little break before the live that's happening soon. So good. Yeah, okay, well, thank you to all our speakers for answering questions, and thank you for everyone that's attending this Q&A and for those that asked questions. And I look forward to seeing everyone at the next session. So we'll end 2 minutes early. Bye, everyone.